So can you hear me? Yeah? Okay, great. It's a pleasure to be part of this session, and thanks for sticking around for the last talk of the day. My name is Ginny Edgcomb. I work here at Woods Hole Oceanographic, and of the various types of interactions between protists and different components of the plankton that my lab looks at, the one I'd like to talk to you briefly about today is symbiosis. Protist interactions play very important roles in the flow of carbon in the ocean, and Alex Worden's figure here does a nice job of depicting this. Protists are important members of the phytoplankton, and as we've just heard about, many are mixotrophs, and together with the heterotrophic protists, they form an important link between prokaryotes in the ocean and higher trophic levels. And through their collective activities, including grazing, they contribute to pools of particulate and dissolved organic matter and to fluxes of carbon to the seafloor. My lab is particularly interested in studying oxygen-depleted regions of the ocean, and this is because these are expanding worldwide. This map of the world oceans by Schmidtko shows you regions of the world oceans in pink that have experienced significant oxygen depletion over the last 5 decades at some depth. And protist-prokaryote associations are common in anoxic and oxygen-poor environments, and this has been well documented in the literature over at least the past 3 decades, primarily on the basis of microscopic observations. And I'd like to give you a couple of examples of these to set the stage for this today. The first is Calkinsia and Bihospades, which are 2 very closely related taxa within a euglenozoan clade symbiontida, which can form fairly dense swarms in low oxygen anoxygen marine sediments, 10 to the 4th per square centimeter, in hotspots of sulfur and carbon cycling. And we've documented these taxa in places like Santa Barbara Basin, Framvaren Fjord, Cariaco Basin, Boundary Bay, Canada. And when we looked at these 2 taxa, Calkinsia on the left and Bihospades on the right, these 2 closely related euglenozoa host autotrophic nitrate-reducing sulfide-oxidizing epibionts related to Archaeobacter. And these epibionts may utilize hydrogen in addition to sulfide as an electron donor, thus enhancing the function of the host hydrogenosomes. Hydrogenosomes are mitochondria-like organelles that protists who live in anoxic environments can use to generate ATP. The host may locate these ectobionts within biogeochemical gradients that maximize the growth of the symbionts. And in these scanning electron micrographs here, the Bihospades on the left and Calkinsia on the right, you can see are densely covered with these epibionts, which are connected to the exterior of the cell by a fuzzy glycocalyx on the cell surface with conduiting through the extracellular matrix. And hydrogenosome-like organelles are situated just inside the plasma membrane. And there appears to be a coevolutionary history between these 2 sets of closely related partners from distant locations. Also in Santa Barbara Basin, there's an abundant ciliate. It's a pigmented ciliate. It's a carrier-relicted ciliate. And this ciliate has inside its body many of these membrane-bound vesicles, which contain non-random physical assortments of various endobionts. And cardfish and ribosomal RNA analyses confirm at least 2 types of sulfate reducers, a bacteroide, a methanogen, and the internal membranes of one symbiont type are reminiscent of a type 1 methanotroph, but we have yet to find more evidence of this. These ciliates can be quite abundant as well, 71 per cubic centimeter in anoxic sediments. In the Cariaco Basin, which is just north of the coast of Venezuela, it's a permanently stratified basin. It's a natural laboratory for studying the microbiology of stratified water columns. And in the Cariaco, we found distinctly different populations or communities of protists in the oxic overlying waters than we found in the oxycline waters or in the deeper anoxic and sulfitic waters. And in the anoxic sulfitic waters, on scanning electron micrograph membranes, almost every ciliate, there were fairly abundant ciliates, and almost every ciliate invariably hosted visible epibionts. At the bottom of the Mediterranean Sea, there are deep hypersaline anoxic basins. You can see one in the background here. They're on the bottom of the seafloor. That's redundant, bottom of the seafloor. That's stupid. Okay, anyway, each of these basins has very unique chemistries. They host unique protist populations, and the ciliates are prominent. They all almost invariably host visible epibionts. So laying that as just a set of observations, it appeared to us that symbiosis may be an adaptation to low oxygen and sulfitic habitats. And in addition to the free-living bacteria and archaea that we all study in the ocean, the collective activities of hundreds to thousands of symbionts per host cell likely contribute significantly to carbon and other nutrient cycling where these hosts are abundant. So we are interested to know how these associations and their activities change along oxygen gradients. So to dig a little deeper into this, we looked at the metabolic coupling between one ciliate, a Metopus we refer to as strain CSS, and its methanogen symbionts. And I should mention that this work was predominantly done by Roxanne Beinart, who was a postdoc in my lab and is now a professor at University of Rhode Island. So this ciliate was isolated from anoxic sediments in the Czech Republic by collaborators of ours, and it hosts dense populations of intracellular methanogens. You can see the characteristic autofluorescence of methanogens here, and it also has surface-associated delta proteobacteria. We wished to assess the activity of this ciliate microbiome using both experimental measurements of metabolism as well as comparative genomics. And for the comparative genomics, we handpicked about 60 cells, we starved them, washed them, we sequenced draft genomes for these guys, as well as for no-cell wash controls. We co-assembled all these libraries. We mapped our reads to bins that were identified as the Methanobacterium symbiont, a heterometopid ciliate, the host, and a bin for other, which we think is largely populated by the sulfate reducer ectobionts. The reads mapping to Methanobacterium were reassembled, and this bin was deemed to be about 93% complete with low contamination. And interestingly, the size of this endobiont genome doesn't show the characteristic genome reduction that one might expect for an endosymbiont. It has slightly higher gene density. When you look at hierarchical clustering of gene counts within each archaeal COG category, normalized to the number of archaeal COG annotated genes in each genome, this endosymbiont genome, highlighted in red in comparison to the free-living Methanobacterium genomes, shows more genes involved in inorganic ion transport and metabolism, amino acid transport and metabolism, and post-translational modification, protein turnover, and chaperones, possibly reminiscent of adaptation to an endosymbiotic lifestyle, but we have to look a little further into this, and fewer genes in transcription and RNA processing and modification. Consistent with the work of Fenchel and Finlay, who for quite a few years studied in culture ciliates with methanogen symbionts, we found that the activities of these methanogens were critical for the survival of its host. So the doubling time of this ciliate is about 57 hours, and when we inhibit methanogenesis with 2-bromoethane sulfonate, the growth of the host effectively stops. We observed on the bench, however, that this ciliate can host a wide range of oxygen conditions. It was quite happy under fully oxic conditions and under totally anoxic conditions. And so we wanted to know how the activity of the methanogen symbiont responded to these different oxygen concentrations. So we measured the production of methane in the headspace of our incubations using gas chromatography under varying dissolved oxygen conditions. This work was done in the lab of Jeff Seawald here at WHOI. We monitored oxygen using an O2-depleted system. All experiments were performed in triplicate. And because this host is a heterotroph and is grown on a mixed prey community, we had to be very careful. We had to conduct all these experiments on whole cultures, on cultures that we had removed the ciliate hosts from, and on whole cultures that we had inhibited methanogenesis in, so that we could look at whether there was methane lost over the course of the experiment. On a per host cell basis, methane production is dependent on the host growth stage. This is one of our key findings, that the average rate of methane production in terms of nanomoles per ciliate per hour was quite a bit higher during the host's early exponential phase as it was in late exponential or stationary phase, suggesting that the activities of the symbiote are very much linked to the metabolic activities of the host. More interesting to me even was that on a per methanogen basis, the rates of methane production were very high relative to documented rates of methane production in free-living methanogens by Caldwell et al. In 2004. These are the rates documented by Caldwell, and you can see that on per methanogen basis, our observed rates are anywhere 5 to 10 times higher than that, and that's based on a conservative estimate of around 500 symbionts per host cell. And I think that's conservative because even though in some cases there were even more symbionts per host, in most cases in the cultures that we had, the number of symbionts was closer to around 200 per host. Oxygen did impact methane production over the 10 to 12-hour incubations. The rate of methane production in nanomoles per ciliate per hour was around 4, and we were not able to detect accumulation of methane in the head space under 1, 2, and 4% oxygen. However, in the BES-treated cultures, the inhibitor of methanogenesis, we noticed that methane concentration decreased with time under aerobic conditions. And we think this is because of the presence of methane oxidizers in the mixed culture. And therefore, methanogenesis just may not occur above rates of methane oxidation in the presence of oxygen. And this is supported by transcriptomes that we ran for each of these experiments, which show that expression of genes of methanogenesis were not significantly depressed under oxic conditions over the 12 hours. So the proposed model for evolution of protist-microbiome interactions in anoxic habitats is based on hydrogen transfer, whereby the host under anaerobic conditions ferments its prey, producing pyruvate, which is taken up by the host hydrogenosome to produce ATP, also producing hydrogen, which is taken up by the microbiome to produce methane or hydrogen sulfide, depending on the identity of the microbiome. And we're in the process of trying to untangle these coupled metabolisms using new Raman approaches. This part is a collaboration with Gordon Taylor at Stony Brook University, whereby we provide stable isotope labeled substrates that are just available to either the host or the symbiont, and then tracking the movement of these substrates through the consortium. To summarize this part of my talk, I just want to say that the activity of protist microbiomes is likely very significant and should be accounted for in our estimates of key processes, particularly in the types of environments where we've documented the abundance of these consortia. This is because the numbers of symbiont cells, which can be tens to thousands per host, may in some cases rival or exceed the free-living counterparts of those same taxa in the water column. And host-provided substrates, niche, and positioning in the water column may increase rates of the symbiont metabolism. Coevolution with the host may also drive selection for elevated metabolic rates. So now I would like to shift to another type of symbiosis, which is parasitism and its possible impacts on the marine carbon cycle. Protist parasites are very abundant. Okay? Parasitism of plankton by eukaryotic parasites affiliated with two groups that are hyperdiverse groups, They're known as the marine alveolate malve groups 1 and 2. In groups 2 is best known by one taxon, the Symbionts, and these may make significant contributions to pools of particulate and dissolved organic carbon where hosts and parasites are abundant. And recently there were 2 very large global ocean surveys, the Tara Oceans expedition and the Malaspina expedition. The Tara sampled the surface waters and deep chlorophyll maximum, and the Malaspina, which I'm depicting in the red dots, sampled the 2,000 to 4,000 meter depths. And in both studies, in their marker gene libraries, these malve groups represented up to 40 or 45% of all of their marker genes. The known hosts include diatoms, radiolaria, ciliates, copepods, and fish larvae. But yet there's still a paucity of data regarding host parasite specificity and infection dynamics. And this is a gap that we're trying to fill now in my lab. The clustering heat map of regression-based modeling of the Tara data, this is a paper by Guidi et al. In 2016, using a hierarchical, okay, so the regression modeling of the Tara data against environmental variables here shows a strong correlation with certain protist taxa and carbon export at 150 meters depth. And in particular, some of the strongest correlations between carbon export were with the alveolate parasites. These parasites have an infectious stage of dinospores here, which go on to infect their hosts, a wide range of hosts. Little is known, however, about host specificity. The parasite invades the host nucleus, grows into a trophont, lyses the host, releasing this weird-looking vermiform stage which dissociates into the dinospores. And in the process of lysing the host and releasing these dinospores, these parasites are contributing significantly to dissolved and particulate organic matter. In Saanich Inlet on the coast of Vancouver, we conducted multi-level indicator species analyses used to reveal OTUs that would co-occur along this stratified water column, and Symbionts MALV2 group OTUs were the most abundant indicator OTUs during peak stratification. As an example, here we have depth from 10 to 200 meters. Here's the oxygen profile that goes along with that. A Phaeocystis bloom is detected in May in green. Okay, all the dots that are not green are Symbionts OTUs, and you can see that the Symbionts OTUs co-occur or track the sinking Phaeocystis bloom over the season. Pick it up again with a couple of weak Phaeocystis blooms a little bit later. Now, a caveat with indicator species analysis and network analysis is that they can't prove host-parasite relationships, but they're great for hypothesis testing. And that's exactly what we're doing in the lab with my grad student, Taylor Sahine, who's in the WHOI-MIT joint program, who's investigating host-parasite dynamics and the impact on the release of particulate organic carbon in a stratified coastal pond here in Falmouth, because it's very convenient, and this allows her to conduct high-resolution sampling up to 3 times a week between March and November, because the parasite life cycle is only a few days. And she will do single-cell sorting. She'll identify with PCR both the host and parasite to gather more data on hosts, the specificity of host and parasite relationships. She wants to generate draft genomes for some of the most poorly understood of these malve clades that we know very little about, but they're found all over the world oceans. And most important, or most interesting to me, she wants to do lab studies of host specificity and of carbon release for the ultimate purpose of modeling the impact of parasitism in salt pond on releases of particulate and dissolved organic matter. And last summer, she sampled salt pond to get some preliminary data. Up to 35% of protists were infected at any one time with these parasites. And the black dots are just the counts of free dinospores, which you can see don't tightly correlate. That's because her sampling was rather coarse. It was once every 2 weeks, and the cycle is much shorter than that. So she would be catching free dinospores after a bloom that she missed. So my closing plea to you is that in your measurements or your estimates or your models of microbial processes and the flow of carbon in the ocean, please don't forget about the protist grazers, the mixotrophic and autotrophic protists, the parasitic protists, nor the symbionts of protists that in some habitats, in many expanding habitats, may play a major role. And many people ignore these or simply place a 0.65 micron filter ahead of their water samples that they collect to measure their favorite process. I call that microbial racism. And with that, I'd like to thank the members of my lab group, past and present, particularly Roxanne Beinart here and Taylor Sahine, who've contributed the bulk of the data that I've presented here today. And I'd like to thank you for your attention, and always remember Gary Larson.
That was fantastic. Also difficult for modeling to do. But in terms of the ciliates with their symbionts, do you know how they control the growth of the symbionts in terms of, normally you'd have them constantly multiplying, usually, with methanogens or other bacteria, their respiration is growth associated. So is there some way that they clamp down on their division rate internally?
That's a very good question. And that's, I don't think, a question that I don't know anybody who can answer that. I have indirect evidence that some of these ciliates have a reduced oral apparatus, which suggests that they may, instead of being true predators, they may actually farm and eat their epibionts. That's one way where they may control the numbers. Yeah, and I do have evidence also with work that I've done with Joan Bernhard here at WHOI, that the same conspecifics in slightly different sites, maybe 50 kilometers away, may not host epibionts, and some do, which means it's driven by environmental conditions.
Thank you.
It's like hot potato.
Hi, Jenny. That was a great talk. My question is also about the ciliate and the endosymbiont. Do you know how tight the hydrogen transfer is, and especially in your oxygen experiments, if you ever see these systems as a transient source of hydrogen leaking out?
Mm-hmm. That's something we're hoping to pick up with the Raman techniques, Allison. I suspect the coupling for endobionts is quite tight. I don't know how leaky the coupling is between an epibiont and the host, but the positioning of the hydrogenosomes right underneath the cell membrane, whereas we usually see them in other locations, suggests to me that it's quite close. I don't know if that answers your question, but I can't put a number on it.
Hi, Jenny. It's
Hey.
kind of related to that question. I'm kind of amazed by the fact that you don't see reduction in the genome of epibiont despite the tight connection. Do you know if it's vertically transferred, or do you see them in the environment?
I think these are vertically transferred, okay? Because, and my only evidence of that is looking at cell division in the lab, okay? You can see the division of the ciliate, and they both come away with endosymbiotic methanogens, okay? That doesn't mean that they can't pick up new methanogens from their environment. I suspect that this particular symbiosis may be one that is more recent, and that's just, I'm pulling that out of the air, but I can't think of another explanation for why the genome has not yet been reduced. Yes?
I'm wondering whether these parasitic alveolates are only found inside living cells or just free-floating in the water, or whether they can also live off of detrital material. Can they attach onto organic particles and grow, or are they only found inside
They need a
cells?
host. But I do, and I'm just a new fan of Symbionts, okay? I was always annoyed by them because they dominated my data sets. But now I'm convinced, like with fungi, that there's something very important going on that I need to pay closer attention to. So one thing I do know from collaborators like Laura Galou in Roscoff is that these Symbionts can overwinter or stay for a long time inside cysts. So you can see them inside taxa that form cysts.
Oh, okay.
So perhaps those cysts get kicked up in the water column, and then they grow. But outside of the host, the dinospore only survives about 2 days.
Oh, wow.
Okay, so if it doesn't infect the host, it just becomes part of the POC.
Huh, okay.
Maybe one more, and then we'll ask everyone to come up.
Yeah.
Thank you for pointing out the importance of parasitism in plankton, because it's hugely important. Do you mind speculating about potential roles for particle flux, export production of maybe infected cells being more susceptible to being incorporated into sinking particles?
Infected cells would be less buoyant, but the sinking rate of infected cells, I think, is going to be much longer than the amount of time it takes to lyse the host. So I'm not sure how, do you follow me? Mm-hmm. I think they're going to lyse their host within 2 days, and the sinking rates, unless you're dealing with a shallow water column, I think are much longer than that typically.
And I wonder if they could be incorporated in or being predated upon more easily if they're more morbid, maybe, potentially being more...
I would think once it's taking over the host nucleus that it would be preyed upon by others. And there are some Symbionts who actually, their modus operandi, instead of attaching to the outside of a host and invading, is actually to be ingested. So some of these dinospores are fed upon, and perhaps if you feed on a host that's infected, depending on who you are. Again, there's still not enough known about the host specificity, so that's something to look into for sure.
Thanks.
That's a great question. Thanks.